Good morning, everyone, and I'd like to thank each one of you for being here to hear out our research findings. So without much ado, I would like to go ahead with my presentation. This is the topic is inter and intra. Is inter and intra-species variation for agronomic, chemical, and anti-diabetic properties of within Ocimum species. We're talking a little bit more about what this is. And I'm the main author here, but most of the work was done by the graduate students. That's what the professors do. They get the grads to do the work, and we present all over the world. Of course, they get the present publications. Dr. Wong and Cantel, they are chemists at USDA, Natural Products Utilization Research Unit. We have a collaboration with them, and they do all the chemical analysis. I'm an agronomist by training, so I grow the crops and send the material to them, and they do the chemical analysis. Then we had Dr. Matthews, he's a professor at Stanford University. He's a nutritional biochemist, and he does all the biochemical essays, part of this project. Project was funded by USDA, and it's my pleasure to present our research findings. So you all know we are herbal products gaining popularity as health supplements in the USA. About 80% of the world's population relies on herbal remedies, one form or the other. And more than 25% of the drugs we see on the shelves are plant-based. Popular one is aspirin we take for headaches and such. Even for the first medicine, Taxol for cancer was actually from the bark of willow tree. So among a wide range of medicinal active plants, basil, particularly holy basil, it used to be called Ocimum sanctum. Later, they have named it as teniflorum based on the angular structure of the stem. It has been shown to have a myriad of medicinal properties and is often considered a panacea for many ailments ranging from common colds to chronic diseases such as cancers, diabetes, and so on. The genus Ocimum belongs to the Lamiaceae family and consists of more than 65 to 150 herbaceous and perennial plant species, primarily distributed across tropical, subtropical, and warm climatic regions. Basically, a tropical origin plant that thrives in warmer conditions. So the members of this plant, Ocimum, genus Ocimum, are collectively called as basil. Known as the king of herbs, basil has a wide variety of applications in traditional system of medicine, dietary supplements, food flavoring, perfumery, pharmaceutical industries, and so on. It's a wide genus that has several species with local and wide adaptations. So it's very well adapted to several conditions. It has been extensively cultivated throughout history, and cross-hybridization resulted in different species, subspecies, varieties, genotypes have been developed over time, and generally not much of significance. This crop, when we plant about 100 different types, and every year we had to have pure seed. There's so much cross-pollination occurs within them that the following year seed loses its original identity. So the advantage here is it's a wide genotypic variation within the species also, and provides an opportunity for potential development of specialized cultivars, which could be utilized for specific purposes and grown in specific environmental conditions. Basil species and varieties have long been recognized as a diverse and rich source of essential oils, which are responsible for both the characteristic aroma and medicinal properties, including antimicrobial, antifungal, and whole range of properties. So coming to its domestication, cultivation, it's been basically cultivated as a culinary herb in France, Egypt, Italy, Hungary, Indonesia, Morocco, Greece, Israel, and the United States. Basil was first introduced in the US in the early 1600s, perhaps from India. American colonists considered basil an essential ingredient in a snuff. They used to take snuff through their nose and considered to ease headaches. They say the people used to work on the railway lines in US. They used to use basil in their snuff and also to keep the mosquitoes off. That's one of the beginning uses in the US. Currently, US imports more than 2,000 tons of basil for its domestic consumption. It's mostly grown in California and Hawaii, and Hawaii is because of the warm oceanic climate, it tends to produce the highest production and also market value in the US. So there are different types of basils: ball basil, short basils, there are Thai basil, purple basil, clove, lemon, different aromas. When we go to the field with about 100 different types, we get different smells. Clove, even cardamom, clove, and such. So I've just given a few pictures of this versatile species out there. So the volatile aroma compounds, they are mainly tannins, gallic acid, chlorogenic acids, etc., alkaloids, glycosides, saponins. It's a very rich variety of essential oils, but major ones would be methyl chavicol, linalool, cineol, and also high content of eugenol. Put a picture here of the glands. These glands are found right on the leaf surface. So that's why as soon as you touch the leaf, they burst and you get a burst of aroma. When our students bring the samples for analysis in the department, the whole building smells of basils. It's a nice smell, and also it's supposed to ease headaches. For purposes of my research, we looked at different species: Americanum, Basilicum, Campechianum, Gratium, Celloid, Teniflorum. We had one from Italy. Later, through chemotaxonomy, we did find chemotyping, found that it's actually a Basilicum. They come from different countries, total about 21 countries. So it gives you an idea how adaptable this species is. Okay. So these are the varieties that species that we used in our research: Americanum, Basilicum, Campechianum, Gratissimum, Teniflorum, and Africanum. So I'm an agronomy guy, so we do planting out there in the field. We raise the seedlings in the greenhouse. They come out pretty quickly, but they grow very slow, painfully slow. What we do is we put organic manures, and then we make the beds like this and cover them up with plastic with drip tape underneath, basically for fertigation. This is organic soluble fertilizer, and planting here at the beginning, and then over time they grow pretty good. Okay, this is our crop. Okay, a student who did all the analysis extraction part of her MS, and there was another PhD student who did the agronomy. So for essential oil estimations, we actually selected about 26 different lines from each of these 6 species. But original germination and such, we did on all the 100 different types. And we saw some variation between the species and also within accessions for germination rates. And you can see here, this unknown basilicum did pretty good. And this celloid was pretty slow. And percentage germination was somewhere between 84.5 to about 100%, some of these lines. Okay. Uh, Looks like this slides moved. Um, anyway, this is the total plant biomass, fresh biomass, and the unknown topped the list. And cell oil was pretty slow. And also, if you look at the fresh leaf weight, also there was variation among the species, mostly between basilicum, teniflorum, and this one is americanum. Okay. So we looked at the range of variation for plant biomass and leaf fresh weight and also dry weights. And a huge variation. If you look here, 619 to 1090 to 14 to 2000 grams per plant. And similarly here, dry weights also pretty good variation among the species and the overall species and the accessions within. Okay. Diameter partitioning also. Pardon me, the slides moved. I didn't know that this will happen here. When I said them, they were pretty good. So dry weight partitioning shows how how efficiently the plant partitions the accumulated dry matter into different plant parts. It's called dry weight partitioning. It's very important when it comes to selection of varieties. If you're looking for the root or the leaf or the stem as the agronomic part, then we want to select varieties that partition more dry matter to that particular economic part. In this case, we'll be looking at leaves. So gratismum tended to be this one here. Gratismum was much better compared to rest from the leaf production point of view. And this inflorescence gives you an indication of how early or late the plants mature. Because when we harvest at the same time all the species or the accessions, the amount of inflorescence gives you an idea whether the plant has already gone ahead into reproductive phase or it is still in vegetative. So gratismum seems to be still in vegetative phase. That explains higher levels of stem and leaf. Okay. We did have some issues with insects. These are Japanese beetles, and they are very sporadic. They come certain seasons and for a short period of time. We just sprayed some neem oil extract, and that took care of it. Okay. So in the first study, we looked at their variation in the, first we looked at the agronomy, biomass and leaf production. Then we looked at the chemical profiles of these different species. So these are the species here, and then these are the major essential oils. We got something like 100, 120 peaks. Out of that, we just selected the few economically important ones. And I've given here in the third column their biosignificance, what these particular essential oils are used, like in the case of piperton, epoxide, fenchone, and humulin alpha, used in bronchitis and the gastrointestinal discomfort. Bergamotin is a major compound for for wound healing and such. And basils are known for methyl eugenol and eugenol, and they seem to have all kinds of properties, antibacterial, allergic, and all this. So myrcene to some extent in celluloid, antibacterial properties. And teniflorum is for the holy basil, also has very rich in isoeugenols and bisabolin and caryophyllene. Okay, and also wide usage in all kinds of medicine. Then we looked at the agronomy, developed this as a commercially cultivable crop. I had a PhD student, Cedric Sims. So what we did here is we selected 3 accessions from holy basil, Ocimum teniflorum. First, we did the planting date trial, 3 monthly plantings, April, May, June. Generally, summer crops, we usually plant in May. We wanted to look if we can get a little early. Season is generally restricted between May and November. because of early frosts in November or late frosts in April. So most of the summer crops are planted in May. So you use the split plot design, main plot, planting density, dates, subplots, so on. Okay. I'm sorry about this. These have gone all over the place. Some of the graphs are missing. Okay. I had fresh biomass. Generally, if you look here, red bar is for the first planting date April, then a yellow one is for May, and this third blue one is for June. So we found that there was varietal variation. The first one is for this variety here, AIMS 23154. This is for the second one, 056, and then 38779. And we found that generally May plantings tended to be optimal. Then we looked at inter-row spacing. The basils are generally planted about 45 centimeters within row and about 2 feet between the rows. But here we looked at 1 foot, 12 inches, 1.5, and then 2 feet. Here we took only one accession, 652056. Okay, this is not bad. So if you look here, as we increase the inter-row spacing, the biomass was higher. So it's kind of, generally we say that closer the densities, you get better yield. That would be on a unit area basis. Space, more resources, makes more branches and so on. We did look at plant height and branches. It was higher in the case of higher inter-row spacing, 24 inches. Okay. And then we looked at nitrogen fertilizers, three doses. There's a control there. Then we had 75 pounds per acre or kilograms per hectare, 150 kilograms, and then 225 kilograms. These were given, this was basal just at the time of planting. Then he gave 2 split doses, and this one about 3 split doses. The fertilizer we used was calcium nitrate. And see here, there wasn't much difference between 150 and 225. Okay. Actually, somehow depressed with more nitrogen here. So as we increase the dose of nitrogen levels, the biomass increased, but optimum appeared to be 150 pounds per acre or kilograms per hectare. So same trend was seen in the case of leaf biomass. We gave fresh leaf biomass because that is the marketable product out there. This is Cedric Sims looking at light interception within these canopies. Next one we looked was mulches, different colored mulches. He had red, green, and I think he had black, and then no mulch. So the biomass varied with the mulch type and also the genotype accession. This is 30 days after transplanting. This is 60 days after transplanting. You can see here that green mulch appeared to be better here. Initially, there were clear differences, but as the season went on, progressed, the differences were kind of reduced. But still, there were varietal differences here. 652057 did very poorly. That's in the control. We look at the green, and this not much of difference, but still little lower 60 days after planting for the total leaf weight and also whole plant biomass. So this is overall, if you look here, mulch color, green, red, green, and control. So really not much for the variety. Ames 23154, 506 here, red one, much depressed in the control. Okay, then we also looked at conventional organic production systems. Again, there's a lag here in the organic. If you see here, yields were reduced, and there were differences between the variety, the accessions here. Somehow 057 didn't do so well, but the others did okay. And with organic, it's called a lag. We get that usually about 15%. Sometimes with proper planting dates and such, you can reduce it to 5%, 10%, but it's always a difference there. Okay. So in summary, there was a variation for plant growth and development for between the varieties and also between the different treatments. Planting date may appear to be the best, and in the case of spacing, for individual plants, probably wider spacing is good to increase their leaf production. But on the other hand, if your overall hectareage, probably low density planting would be better. High density planting, sorry. These are conclusions. Gratismum did about the best. Then I went ahead. I sent my student to Rutgers University for doing the chemical analysis and did some GCMS. These are the conditions, okay, used for GCMS. So there was a wide variation in the essential oil percentage, total volatile oil. So this is the essential oil expression by basil is controlled by the environment. Okay, species and environment, of course. So here in the beginning, it was eugenol with at 60 days. Look here, same variety, same variety is the same. You see, caryophyllene was high. Caryophyllene continued to be higher at 90 days also. So eugenol tended to catch up here between the planting, between the different varieties, and there's no significance between them. Yeah. And this is the other variety. Same, there were differences in the beginning, and then if you see here, eugenol, it expressed in June, 60 days of planting, it was higher there. And here it is, again, not much of a difference. Different chemical was up there. Okay. So in essence, there was a difference in the type of essential oil that was expressed due to time of harvesting. So initially we had eugenol, then we also had, in the case of this variety, beta caryophyllene was higher. And in this case of accession 779, beta caryophyllene was a dominant oil, but the percentage content decreased as the plant matured. And then eugenol tended to increase with time in April, but decreased in May and June. So each here, this gives you an idea of tailoring and planting these and harvesting them for particular essential oil. Okay. Spacing also had an effect on these varieties. If you look here, 45 centimeter spacing. Okay. Yeah, this one. Okay. This is bisabolin. And here eugenol was higher 60 days after planting. Eugenol continued to be higher here. But if you look at the spacing differences, this blue orange and green color. There have been differences in those different oils at different times, but mostly not significant here. But then there are some slight differences here for eugenol. Okay. So spacing did have some effect on the essential oil composition of the basals. Okay. This is levels of nitrogen. We just looked at 30 and 60 days after planting. And these are the doses that were used: 0, 75, 150, and 225. And again, you see some variation in the oil, essential oil that is dominating depending on the fertilizer dosage. Okay. This is a conventional versus organic. You didn't see so much of variation if you look here, almost similar levels, except for eugenol, but again, not different. So the production system didn't seem to have such a profound effect on the essential oil composition, but only the yields. Same thing here, different variety, 602057. Okay. If you see here, beta carotene tended to be higher in this case. Yeah. Overall, eugenol and our isoeugenol is main component of this. So this is a different trial we did again to look at the anti-diabetic value of this plant. And there was some variation between the species and also the accessions. These are the accessions here under each species. And there's much variation in the plant height, branches, and above ground biomass, and also leaf biomass. Basilicum is generally used for culinary purposes, Italian basil, Thai basil, and all those. But we found that basil, the culinary basil appears to be more susceptible to insects in our environment. Disease, we didn't see much at all. Okay. So the oils that were, we actually here what we did is based on the literature where a particular essential oil is associated with anti-diabetic value. We focused on those oils. We didn't do the whole profile. Earlier, the student did the whole profile. In this case, we just did the this group of essential oils. Okay. Mainly, we focused on rosmarinic acid and also caffeic acid. These were highly good candidates associated with anti-diabetic value. So in our case, we found epigenin, genistein, and then lutecolin. Okay, these three peaks here. It's then show properly. Okay. Okay. Here, these were the flavonoids, phenols, and terpenoids we looked at. The earlier slide showed that. And in this case, we found that rosmarinic acid here, very high, 360. And also among flavonoids, vicinin was pretty good. Terpenoids, most of these betulinic, oleanic, and ursolic acids were high. So this is looked at the anti-diabetic value, looked at the percentage inhibition of alpha glucosidase uptake in the musculoskeletal cells. These are cell studies. And here we used acarbose, which is a common anti-diabetic medicine, and looked at, this is a study that was done at North Dakota State University, where we looked at just the different types of basils, okay, not the original species, but most of these are polybasals. These that have MSR, these are all O-tenuiflorum lines. And we also looked at Thai basil and lemon basil, popular ones, red basil. We also threw in mint. Mint is also part of Lamiaceae family. So it had much higher inhibition, okay, compared to Acarbose, okay. Here we found this variety here, MSR 198, 97, 95. I looked at undiluted, half diluted, 1/5 diluted. Generally, undiluted extracts showed higher activity compared to the diluted ones. Okay. So the reduction was somewhere between 12.84 for amylase, and for alpha glucose, there's 41 to 94% in the undiluted ones. Okay. In the other study where we looked at all this 26, out of that we selected, I think, 16 accessions belonging to those 5 species. And this dark one here is the medicinal Acarbose. And if you look here, percentage inhibition is about 100. Among these, this is Gratissimum line that had pretty high, 80% or so inhibition rate. Okay. These two are, I think this is tenuiflorum line, and this is also tenuiflorum. So all of this, we didn't see any effect on the inhibition of alpha glucosidase. So we looked at the 2 rosmarinic and caffeic acid trends, and as the concentration increased, the inhibition also was higher. If you look at the Acarbose, the trend was about the same. This is for Acarbose percent inhibition. This is the Gratissimum line, which also showed similar trend. And look at their chemical composition, rosmarinic and caffeic acid, same trends here. So we concluded that rosmarinic acid and caffeic acid are good indicators for anti-diabetic value of these basals. Okay, same thing. So in summary, basal field-grown plants, we looked at their chemical composition, mainly those chemicals that are associated with anti-diabetic value, and looked at the anti-diabetic assay using musculocellular cells and uptake, inhibition of glucose uptake. And the best variety appeared to be Gratissimum. Came as a surprise. Generally, polybasal or tenuiflorum is considered as the candidate for anti-diabetic, anti-cancer and such. But in this study, Gratissimum variety from Zambia, it turned out to be the best. Okay. And these 2 essential oils, caffeic acid and rosmarinic acid, were very closely related to anti-diabetic activity in basals. Okay. I'd like to thank you all for giving me an opportunity to present our research here. Open to any questions. Time-wise, I think